Doug Leith, if I remember correctly, you were at one of the very first World Vegan Days going back, I think probably 2004 or something, maybe going back to that era. So maybe even earlier. And I've known Doug for a long, long time, and he's one of the hardest working animal activists that there is. And when it comes to vivisection, there's nobody, I would say, in Australia, maybe in the world that knows and researches the subject as well as Doug Leith. So, and it's a shame that he's on as late in the day as it is, because there's not that many in the audience. But I definitely, those that are here, I recommend that you stay and listen to Doug's presentation, because the information that Doug presents is of the highest quality, and it's very, very well researched, and he really knows his stuff when it comes to vivisection. So without any further ado, I'll bring onto the stage Doug Leith.
Thanks very much to Mark Dondu, who's a very knowledgeable person on veganism and all issues related to veganism, and has been a great supporter of it. I became aware of animal experimentation when I was 18. I went to the Healthier Living show and I saw film of monkeys with their brains exposed and electrodes in their brains. I found that very upsetting. I won't refer to what's actually happening to animals again during this talk, but I just wanted you to know that that was the first thing I saw about it. And I was going to keep on walking and pretend I didn't see that, but on that day I decided not to, and so my life changed, as I guess all of ours did at some point when we found out what's happening to animals on this planet. But for the first few years of my involvement, I did think we only had what is called a moral ethical argument, which is an animals versus humans argument. And while I regarded that as being legitimate, because I don't believe we have a right to do those things to animals, even if humans benefit, it did leave me floundering when someone would say, but if your mother had cancer, would you still want to save a rat? Or a question of that nature. Because I believed that choice existed, otherwise why would they do it? So it was both pleasing to become aware, but also quite upsetting to find out that we'd been very much deceived in that respect. This was not an animals versus humans issue, it is animals and humans versus some very powerful vested interests, inertia, and other very anti-scientific and inhumane practices. Throughout history, animal experimentation has delayed human medical benefit. For example, the creators of penicillin, Sir Alexander Fleming, said, "How fortunate we didn't have these animal tests in the 1940s, for penicillin would probably never have been granted a license, and possibly the whole field of antibiotics might never have been realised." The other two Nobel Prize winners for penicillin echoed that. Penicillin has now saved millions of humans. Dr. Karl Landsteiner, the creator of blood transfusions, also said that only in the highest anthropoid apes do we find things like blood groups that are similar to humans. So, of course, human blood groups could never have been identified through animal experiments. And you need to know what human blood groups are in order to transfuse human blood, and blood transfusions have now saved over 1 billion people. Animal to human blood transfusions even occurred back then, which were, of course, a failure. The creator of the oral polio vaccine also said prevention was long delayed by the erroneous conception of the nature of the human disease based on misleading models of the disease in primates. So the polio vaccine was delayed by 29 years because primates gain polio via the respiratory system and humans via the digestive system. With penicillin, the delay occurred because it was systemically ineffective in rabbits. That means it was ineffective in the bodies of rabbits. It also caused birth defects in rats. It killed the cat that it was injected into at the same time it was injected into a human. It's really just a matter of good luck that we have antibiotics at all. And this is the case across the board. The director of the National Cancer Institute said, "The history of cancer research has been a history of curing cancer in the mouse. We've cured mice of cancer for decades and it simply didn't work in humans." Others have said, "We've cured enough mice of cancer to fill several New York City subway systems." And so on. Not just the case with these diseases, but with any disease you can think of. Humans and animals only get the same diseases 1.16% of the time. Yet almost all human diseases are modelled in animals which don't get those diseases. There have now been over 100 AIDS vaccines that work in monkeys. None of them work in humans. over 150 stroke therapies that work in animals. None of them work in humans. This is occurring constantly, yet the public doesn't hear about the big picture. We only hear about the occasional breakthrough in inverted commas, and we're told that within 3 to 5 years there'll be some benefit to humans. And there'll often be some images of mice in the news story. So what we're left with is a general impression that some sort of advancement is occurring without any actual evidence of it. Cures never coming about, decade after decade. But animals are certainly being cured all of the time. So what are the real motivations for so-called animal research or animal testing? Well, the provision of legal protection to industries who know quite well that their products are harmful to humans. It seems to be the driving force. For example, warnings were kept off cigarettes for 30 years by so-called animal tests. Human evidence already showed that smoking caused lung cancer. Dr. Clarence Little, an animal experimenter at the time, said the failure to induce lung cancer in all but a handful of cases cast serious doubt on the validity of the cigarette lung cancer theory. That was because animals weren't getting lung cancer. So this was a way for the tobacco companies to ignore the human data, which showed quite well smoking causes lung cancer. Similarly with ionising radiation, warnings kept off for 40 years. With asbestos as a carcinogen, warnings were kept off asbestos for 70 years by so-called animal tests. So there is some health being protected by animal tests. That is the financial health of industries who know quite well that their products harm humans. This is the pharmaceutical, the petrochemical, tobacco, and other industries. So that is the first way that animal testing harms humans. It passes as safe substances that are not safe and significantly diminishes the chances of successful litigation of people damaged by those substances. Secondly, it harms humans because it constantly fails to cure human diseases while constantly drawing in money via donations and government tax money, I suppose. So what we're really up against here is not finding alternatives to animal testing. As Professor Pietro Croce, the author of the book Vivisection or Science, A Choice to Make, had to say, quote, "Are there any alternatives to vivisection? Certainly not, because anything intended to replace it should have the same qualities, but it is difficult to find anything that is, and always was, more deceptive, bogus and misleading for biomedical research than vivisection. So what we should speak of are real scientific methods. They are not alternatives," end quote. So I advise you not to use the word alternatives. You're either saying then that the animal tests work or that the scientific methods don't work. You may wonder, why am I even talking about human health? You're not interested, perhaps, primarily in human health. You're interested in helping animals. The public doesn't care more about mice and rats than they care about their mothers and fathers, and they never will. And the vivisectors know that, and they want this to be presented to the public as an animals versus humans issue. We will never win that way, and they know it. The public needs to believe they've heard both sides of an argument in order to make up their mind. And what they will believe is one side wants to save mice and rats and also some dogs, cats and monkeys, and the other side wants to cure cancer, AIDS and every other disease under the sun and protect us from harmful substances. Very few people will choose to save the animals when presented with this choice, when their own family members will suffer from diseases such as cancer and heart disease, diabetes, and so on. So to be effective anti-vivisectionists, we need to realise, firstly, how strong our argument is. This is not an animals versus humans issue. Human diseases are not being cured by animal experiments. And they're not going to in the future. We went our separate ways from mice 60 million years ago and from non-human primates 6 million years ago. Nothing is going to make them predictive for humans in the future. It's impossible. Even identifying the mouse genome and identifying genes that are responsible for cancer or other illnesses in a mouse is of no value to humans, because the exact same gene in a human usually has a completely different effect. Genes can be turned on and off and perform completely different functions between different species, and they do. And there's nothing that can be done about that. Knowing everything about a mouse genome tells us nothing about humans. So there is nothing that can be done to make animal testing predictive or useful for human medical research. This isn't going to change. So be empowered by that awareness rather than being discouraged by the enormity of our opposition. That is what I would like the vegan and animal rights community to take away from what I'm saying and to take away from the evidence. That we have a very strong argument, just as strong as the argument for the vegan diet. We know we don't need to find some alternatives to meat and cheese and eggs. You just don't need them. In fact, you're healthier without them. Well, we don't need to find an alternative to basing human medicine on mice and dogs and cats and pigs and chickens. It's completely unscientific and has no value to us. Real scientific methods exist: microdosing for the testing of pharmaceuticals, microfluidic chips, computer models, and old methods such as epidemiology, clinical research, the observation of cadavers, etc. We don't lack for real scientific methods. But unfortunately, what we're really up against is a very mercenary opposition who seeks legal protection and will present it to the public as a scientific rather than a legal device. As a former scientific director of Huntington Life Sciences said, the best guess for the correlation of adverse reactions between human and animal toxicity data is somewhere between 5% and 25%. In other words, the animal data is only correct 5% to 25% of the time. This is on a whole cross-section of species. In regard to teratogens, that is substances that cause birth defects, it was said of 800 substances in our database known to be teratogenic in animals, only a small number of these, approximately 20, are also teratogenic in humans. This difference can be attributed to metabolism, exposure time, and species difference, end quote. That means a failure rate of 97.5%. And you'll find this is the case across the board. Nothing is more than 50% accurate when it comes to animal tests. So the evidence is on our side. It's not going to change. Please use this evidence. Go to websites such as safermedicines.org, Medical Research Modernisation Committee, Campaign Against Fraudulent Medical Research, Europeans for Medical Progress, Americans for Medical Advancement, Humane Research Australia. Or you can speak to me directly through Animal Liberation Victoria, or contact me on Facebook. I'm happy to help anyone with this information. We have a strong argument. Thank you for caring about this issue, and I encourage you to be empowered by this information and to get this to the public. Thank you. In regard to other areas, the Drey's eye irritancy test you may have heard of, in which rabbits, usually unanesthetised rabbits, have corrosive substances put into their eyes. A random test of 14 substances when compared to human data showed the rabbit data to be inaccurate by a factor of 18 to 250. A factor of 1 is 100%. That means the closest the rabbit data came to being correct for humans was being wrong by 1,800%. And the most inaccurate of those 14, it was wrong by 25,000%. We don't need to find alternatives to this. It doesn't work. Nothing's going to change. There are fundamental differences between the human eye and the rabbit eye that make that data useless. Rabbits are used because they have big eyes, and they're cheap, and they're docile, and that's it. So it's all about practicalities and expense and making things easy for the vivisector. The same reason that beagles are used. Very tractable, as they'll call it. Otherwise, they won't bite you. Nothing to do with predictiveness for human beings. Even primates, non-human primates, the amount of DNA the two things have in common is not proportional to their utility for any practical purpose. As I said earlier, there's been over 100 AIDS vaccines that work in monkeys. None of them work in humans. They don't get hepatitis, that malaria, other diseases that humans get. But research is still done on these animals for those diseases. Not only has AIDS research in monkeys been a complete failure, but in the French blood scandal in the 1980s, thousands of human haemophiliacs were given HIV-infected blood because it was given to monkeys and it didn't cause AIDS. In Melbourne, you've got all of the universities. Then you've got Walter and Eliza Hall Institute. Then across the road, there's Rotary Bone Marrow Institute, the Howard Florey Institute. Then up the road on the left, this is Royal Parade I'm thinking of. You've got the Monash pharmaceutical place, so that's just within about 2 kilometres there. You've got CSIRO. The Baker Institute uses large animals. They use sheep and other animals for heart research, which probably means they either induce stroke or heart attack in those animals, which has nothing to do with human stroke or heart attack. Animals are not appropriate models for human stroke because they have what's called a returnable circulatory system in their brain, which means blood clots bypass the brain. In humans, this doesn't happen. This is a fundamental reason why stroke research in animals is of no value. There are different reasons why animal research is of no value for different diseases and species, but it is the case almost across the board. Only 1.16% of human diseases are also shared by other species of animals. And this is a problem which cannot be accounted for or dealt with. Some would say then, but what about basic research? You do this on animals, and maybe sometime in the future that thing that worked in animals may turn out to have an applicability for humans. Well, when 30,000 examples of basic research were given 30 years to translate to human benefit, and they're not all animal experiments, but the majority are, only one turned out to be of use to humans, 1 out of 30,000. That's ACE inhibitors, angiotensin-converting enzyme inhibitors. But ACE inhibitors are the result of rational drug design. They're designed with a pencil and paper. They're not the result of animal experiments. So they scored 0 out of 30,000. And the same is the case across the board. The National Cancer Institute was forced to admit that after testing 400,000 substances on mice for anti-cancer effect, they didn't produce any that helped humans. That was over a period of 30 years. They only stopped doing it because they were taken to court. And in court, they actually had to say they were trying to cure cancer in mice. Yes, animal testing is increasing. There's about 305,000 pharmaceuticals on the market. That's increasing. They're all animal tested. Interestingly, a publication called Innovation or Stagnation, Challenge and Opportunity on the Critical Path to New Medical Products, US FDA 2004, said 92% of pharmaceuticals fail human trial after they've passed all the safety tests in animals. So they acknowledge that the animal tests are wrong 9 out of 10 times. What we need more there is microdosing. Microdosing means you put a small amount of the substance into a human who is suffering from the problem. Prior to that, you test on a microfluidic chip, then a computer model, then into the human. This actually diminishes the pain, the injury to humans, and makes safer pharmaceuticals. What we're really up against is that, according to the World Health Organization, 99.7% of pharmaceuticals cause side effects. They can be as minor as a headache, but they can also be as severe as death in the long run. So an industry whose products cause side effects 99.7% of the time would be very foolish to use a test which identified that fact. And that's what microdosing would do. So we have tests that work, but what we're really up against is an industry who does not want to use them, but will continue to give lip service to saying we would love to replace animal tests. We need the public to know that, so they demand it ends. It's not a matter of a lack of so-called alternatives. It never was. Thanks for that question. I'll mention diabetes briefly because a lot of people have it, and Ingrid Newkirk gets criticised because, you know, she takes insulin. Well, insulin was delayed by decades by animal tests. Even Frederick Banting himself acknowledged that it was human studies that made him aware that the pancreas was the organ responsible for production of the anti-diabetic substance that later became known as insulin. It was work by Dr. Moses Barron, called the Islets of Langerhans in relation to diabetes, with special reference to cases of pancreatic lithiasis, which showed that humans whose pancreas had been damaged developed diabetes-like symptoms. Autopsy studies showed the same thing. The work of Zulza and others also showed that people whose pancreas had been damaged developed diabetes-like symptoms. That was all human data. Frederick Banting's work on dogs did nothing to prove that. And his belief that it was true came entirely from those human studies. And the substance he produced from dog pancreas was, to quote Collip, one of his colleagues, absolutely useless for continued administration to the human subject. And even then they tried 12 different species of animals until they found one, the ox pancreas, whose substance was useful for humans. So it's one failure after another of animal experiments, yet they take credit for it and say we only have insulin because of animal experiments. If you have trillions of dollars behind you, you can say whatever the hell you want on this planet. And they do.